Hello, everyone. My name is Gert Lawler, and on behalf of the Foundation for Conservation of Atlantic Salmon, I would like to welcome you to today's webinar. If you wish to use it, we do have AI simultaneous translation available. In the chat, you will find instructions on how to connect audio or subtitles. For today's webinar, we are very pleased to be hosting Cindy Breaux. Dr. Cindy Breaux is a research scientist with the Department of Fisheries and Oceans in Moncton. She's originally from New Brunswick and completed her PhD in 2011 at the University of New Brunswick. Cindy started working at DFO in 2008. Her research focuses on understanding the drivers affecting the survival of diadromous fish to aid in conservation efforts and inform fisheries management. She is leading and collaborating on Atlantic salmon studies spanning North America and throughout the species range to understand factors involved in the declines. Today, Cindy will be speaking on marine scale growth patterns of Atlantic salmon in the North Atlantic. After this afternoon's presentation, we will be opening the floor to questions and answers. Now I will turn the webinar over to Cindy. Welcome, Cindy.
Thank you. First, I would like to say a thank you to the Foundation for the invitation to allow me to present this work. So today I'm going to present one of the projects that we're working on. And for this project, we're using salmon scales in order to look at growth patterns in the marine environment. So several marine ecosystems are experiencing a reduction in productivity, and these are often linked to changes in sea surface temperature, changes in abundance, composition, geographical distributions of plankton and fishes. Now, we know that many migratory fish utilize different marine habitats and may be impacted by these changes in productivity. When we look at the Atlantic salmon, they leave rivers in spring for their ocean migrations in this area, which is the area of eastern New Brunswick, more specifically in the area where the Miramichi River would be. The salmon leave the river, migrate through the southern Gulfs of St. Lawrence to migrate to the Labrador Sea, where they feed, grow, and put on the bulk of their weight prior to returning to rivers to spawn. For each cohort, a proportion of the fish will remain at sea for 1 year. We call them 1C winter fish. The remaining proportion will stay an additional year, so 2 years at sea, and we refer to them as 2C winter fish. And for this presentation, I will be focusing on 1C winter salmon and 2C winter salmon. Here is a figure produced by the ICES working group where it estimates the number of salmon in the ocean prior to any fisheries removal for the time series going from 1970 to the current. And what we can see is that the estimated number has declined over the time series for the North Atlantic overall, for European stocks, as well as for stocks in North America. That has led to many questions about what are the drivers of the abundance, and that has led to different hypotheses. One of the hypotheses that is of interest is the following: that there may be a change in growth patterns of salmon when they're in the ocean, and that perhaps that pattern of growth has changed over time. So how we can tackle that question to look at changes in growth rate is by looking at scales, because there exists some scale archive collection that allow us to look at potential growth. So what we can do, very similar to what's on a tree, is that we can count the circle eyes on a fish scale, similar to what would be done for a tree. So we have areas of more favorable growth conditions, and we have really a lot of spacings between the circle eyes, which are the circle eyes are the lines. So when conditions are favorable, we have a lot of spacing between them. And in the winter time, when conditions are not as favorable, we get really a tightening of the intercircle spacing. So that allows us to go back in time because of the scale archives and actually look at potential changes in growth. We also know that the size of the scale is linked to fork length. There is variability in that relationship. However, using scales can really help us make inference about the growth of the fish. So what do we know about growth in salmon? So there's been a lot of interest in the growth in that first year at sea, so the year of the post-smolt year. And this is a study that was done on Norwegian populations where they looked at that growth at that first year at sea. And they've noticed a sharp decline following 2004, and they've attributed this decline to a change in marine, in a regime shift in those populations. A follow-up study looked at the diet and the stomach fullness of post-smolts from the area. So they compared the region, they compared the time period 1995 to 2004 to the one of 2008 to 2019. And what they've noticed is that there was a decrease in the stomach fullness of post-smolts as well as a change in diet. More recently, there's a study that looked at growth in salmon from rivers in France, and they've noticed as well a similar decline after 2004 in the growth of these fish. So a similar trend to what was found in Norwegian rivers. In the meantime, here in North America, we were also measuring salmon scales to try to get a handle of what's happening in terms of growth and if we can detect any changes over time. So the overarching question of this project is really to look at growth patterns of returning salmon in North America and try to determine if we're seeing similar patterns than what they're seeing in Europe. So more specifically, the first question is to look at temporal change in scale characteristics over time and try to determine if they differ for 1C winter and 2C winter fish, because within a cohort, there's not, to our knowledge, there's no comparison between the two age groups to try to see if they're experiencing similar conditions or not. The second question, can we use scale characteristics to try to predict what the fork length of fish returning to rivers are, which would give us an indication of whether there's particular periods of time on the scale that would be linked to fork length when fish return to rivers. Lastly, we were interested to determine whether there's correlations in scale characteristics between the two age group within a cohort. So that would allow us to kind of infer if they are experiencing similar growth conditions at sea or not. And when I make reference to that third research objective, it really is focused on that first year at sea, because that would be the time period when the two age groups would be out in the ocean. So how we did this, so we used an archive scale data collection that was gathered by the DFO science team on the Miramichi River. A lot of work has been put into collect over the years to actually gather that data set, so I wanted to point that out. What we're doing is using scales in order to infer marine growth at different periods of time. So we're looking at the scale over time and trying to see if we see any changes. To do so, we measured 50 scales per age group per year over the small cohort years 1958 to 2016. This is one of the scales that we measured. It's a 1C winter that was caught at the DFO trap net on the Southwest Miramichi River. It was caught in 2005. Scales were taken from that fish. So we took images of the scale, drew a transect, and measured all of the circuli, so the black lines, on the scale in order to measure growth at different time periods. So that allows us to measure growth in the freshwater environment. Here is the marine entry, so when the fish first enters the marine environment. So we can measure from marine entry up to summer maximum. And summer maximum is going to be of interest later because it's the point in the summertime where the highest growth occurs. I'll also be talking from the summer max up to the winter min as well. So those will be different I will present data on these different sections of the scale. So the first question, are we observing any temporal change in scales characteristics between the two age groups? So here is a figure of fork length in relation to smolt cohorts 1958 to 2016. The black symbols are the 1C winterfish, 2C winterfish are in yellow. And what we see is that there's a significant increasing trend over time for both age groups. We fitted a GAM model shown in green and also a generalized linear model in pink. And what we see is that there's no difference between the age group, meaning that there is a significant increasing trend. However, they're the same for both age groups. When we look at the relationship between scale length and year, same pattern as we're seeing for the fork length. So we're seeing an increasing trend, no difference between the two age groups. Now, we are focusing today on the first year at sea to try to see if we're seeing a similar trend than what was seen in Europe. So if we look at the same figure, but for marine entry up to the summer max, so this is the time when the fish enters the ocean up to the maximum summer growth, what we see is that there's not a significant relationship between over time, and the models indicate that there's no age differences either. However, when we look at growth from summer max up to the winter period, what we see is that there's a significant increase over time, and that's the same for the two age groups. So there's no difference between the two age groups, meaning that they are both increasing over time. And that actually translates into the size attained at the end of that first year at sea. So what we're seeing in recent years is that there has been an increase in the size attained by the fish of both age groups, so such that there is higher growth in recent years. So this is very different than what they're seeing in European populations, while they saw a declining growth in after 2004. We're seeing the opposite. We're seeing an increase in that first year at sea. Now, the second question we were interested in, can we take scale characteristics, so specific period of time on the scale, and can we link it to fork length of returning fish? Before I present the information, I wanted to show you one example. So this is an example of fish in 1 year. So we have 50 fish that we measured. These were 2C winter fish. We plot fork length in relation to plus growth. So I'll show you on the figure what plus growth is. So for 1C winter fish, it would be from the minimum during the winter to the edge of the scale. For 2C winter fish, it would be during the second summer maximum to the end of the scale. And what we see is that there's a lot of variability in the individual data points. And what we and what we're doing here is really fitting a model to try to get at the overall relationship. So we ran linear relationships between each of these scale characteristics. So we have total scale freshwater zone, so the growth in the freshwater zone. These three are growth during that first year at sea. So this is the time when that first year people are really focused on, because they think that it's an important time during the growth period. And for the 2C winterfish that spent 2 years at sea, we have growth during the summer, the second summer. We have plus growth for the 1C winterfish and plus growth during the second year at sea for the 2C winterfish. Not surprising, for both 1C winterfish and 2C winterfish, the total scale, the length of the scale, is a good predictor of fork length of the fish when they're returning. We also see that for 1C winter, the freshwater zone is also important. What's interesting here is that growth during that first year at sea, it's not different than 0 for 2+ or for 1+. So it doesn't seem to be a good predictor of fish, of the size of the fish returning to the river. What's interesting is that for 1C winter, surplus growth, so growth that's put prior to migrating back to the river after that, after the winter, is an important predictor of size. And that's the same for 2C winter during their second year at sea. So the plus growth that they put at the end of the scale is an important predictor of the size of the fish returning. The last question that we will be looking is trying to understand if there's correlation between age group in any of the scale characteristic to try to understand if they're experiencing similar growth conditions at sea or not. So the first response variable that we look at is growth. So just the linear distance of growth on a scale. So here we have within cohort, the growth of 1C winter in relation to 2C winter. It's a straight correlation. And what we see is that there is really a strong correlation between the two age group within a cohort. When we look, and that's for this upper graph is for marine entry up to the summer max. When we look summer max to winter minimum, we see a same pattern. And when we look at growth, overall for that first year at sea, again, we see a really strong correlation. When we look at the difference between the two age groups in comparison to the one-to-one line, we don't see any temporal trend. There may be some outliers here earlier in the time series. However, they're evenly distributed between up above and below the line. Now, so we see a strong correlation when we look at the distance. What about if we're looking at intercirculi distance? So intercirculi distance is really that distance that we just saw, but we divided by the number of circuli, which is termed the intercirculi spacing. When we look at that same relationship within cohort, there is still a significant relationship. However, it's not as strong as what we saw earlier with the growth. And we see a similar pattern when we look at different regions of the scale. So we see that there is some significance, however, it's not as pronounced as what we saw earlier. When we look at the difference between in relation to the one-to-one line, I'd like to point your attention to those two graphs here. So what we are seeing here is that the outliers are all gathered below the one-to-one line in both situations. So this is starting from the summer max to the winter minimum, and for that first year at sea overall. And this is systematic bias towards below the one-to-one line. And what that is indicating is that it's indicating that growth of 2C winter is consistently less than for 1C winter salmon, which is supporting evidence that there may be a link between age at maturity and growth. So such that 1C winter salmon are maturing earlier, but they have a higher growth compared to the 2C winter salmon. Now, can we use that intercirculi spacing as evidence of similar growth condition? Here is a plot of intercirculi spacing. So that distance over the scale, over section of the scale in relation to the number of circuli, in relation to marine circuli number. So this would be on the scale. So this would be the circuli of marine entry, the second one, the third one, etc., all the way to the end of the scale. The green dots are observations for one fish. So this is one marks that were put on the scale for one fish, just to show the variability. And what we did is we did a model specific for each individual fish, as well as a global model for all of the fish within an age group within a year. And we did that because we're trying to get at overall patterns of interest and try to not look at the noise that may be in the data. So that would help us to get at whether we see similarities or not between age groups. A different way of presenting this data is really that each of these gray lines are individual fish within an age group within a year, and then we ran a global model to get like a smooth of the distribution within an age within a year. And that's really going to help us to compare the two age groups within years. So what we're presenting here is the same figure. So we have intercircularized spacing, we have marine surplus number, and what we decided to do is really show the data in jumps of 5 years. So we have 1958 here, 1963, 68, etc. In orange are the 1C winter fish, in blue the 2C winter fish. So this is looking only at the smooths of the model, so to try to see how they compare within cohort between age class. And what we see for most of the panels is that there's a lot of overlap within a year. So for example, in 58, we have an initial increase in growth, and then a small decrease followed by an increase. We see that in both groups, the 1C winter and 2C winter of that year. We see afterwards a decrease to the winter minimum, and then we see an increase again. And this is where the separation occurs between the 1C winter and 2C winter fish. So you start to see an increase in growth, followed by a decrease in 1C winter, because these fish are stop growing and return, they're on their return migration to the river, whereas the 2C winter remain an additional year in the ocean to grow. And this pattern is really consistent in most years. There are some years, for example, in 78, where they don't seem to be doing the same thing. So we have some separation between the growth of the two groups. But overall, we do see that if there's one peak, it's the same for the two age group, followed by the decrease. And there's a paper by Carlson and al. that attributed the winter annulus to be towards the end of February up to mid-March, which would put it somewhere around here. So, so if we rely on that study, it may be suggesting that the separation or the divergence between the two age group could be occurring sometime between April and May. So that would be a logical date that would make sense in terms of the two groups separating within a year. So this plot is similar to the previous one, except that we put year of smolt migration on the y-axis. So we have the cohort 1958 all the way to the cohort 2016 here in relation to the marine circuli number. So here is marine entry, and then we see that summer peak, and then that winter minimum. And what's interesting to see about these patterns, this panel is for 1C winter and this one for 2C winter, is that there's a lot of similarities between years within an age group. So we have like a maximum here that occurs in the summer period, followed by a winter minimum. And like I mentioned earlier, we see here a pickup in growth, so the fish start to grow again, and then we get that separation where the red dots are occurring. So the red dot indicates when growth picked up, and then you start to see less growth in the 1C winter fish because they're returning, they're on their return migration. So now that I've covered the material, I just want to recap. So to say that we are not seeing the same decrease in growth in that first year at sea. If anything, we're seeing an increase in growth over time. When we look at the cumulative growth overlap between the two age group within age class, we are seeing a high level of correlation, which points to factors at sea that could be acting in a similar way on the two age group within cohorts. So this could be food or temperature. So when we look at the two groups and where they diverge, so we see that there's really similar patterns. And then at one point at about circuli 30 to 40, we see that the fish start to diverge such that the 1C winter are returning and the 2C winter salmon are continuing to their growth in the marine environment. With that, I would like to acknowledge the DFO science team on the Miramichi who has been collecting this data and those samples for many years. Without those samples, we wouldn't be able to conduct such a study. And also Colin and Jill for actually leading the measurements of the scales. I would like to mention that my email address is here on the page. So if there are any follow-up questions, please feel free to contact me. With that, I'd be happy to entertain any questions.
Thank you, Cindy. That was an excellent presentation. We will now open the floor for questions. Please raise the little hand on your screen and I will be able to unmute you so that you can ask your question, which is our preference. But if this function does not work on your device, you can type in your question and I will read it out loud. Also, a friendly reminder that we have limited time, so please keep all questions on topic. So we'll just wait to see if there are any questions. Okay, Bill Haley is about to ask a question. Bill, you can go ahead.
Okay, thank you. Bill Haley from Marguerite Salmon Association. Cindy, was there any division in the sex of the fish? Especially I'm looking at the 1C winter. Here in the Marguerite, just about all of our girls are male. Is it a similar situation in the Miramichi? Was the sex of the fish recorded?
Yeah, it's a tough one recording the sex of the fish because especially before fall, like it's really difficult to identify if it's a male or a female. For another project we were looking into, we looked at whether we had enough data on the sex of the fish, and it wasn't there wasn't enough. Just because of that uncertainty, it is expected that for the Miramichi would be similar to the Marguerite, so the majority of the grills would be, no, I say that's not the case. So for grills in the Miramichi, we do have data on male and female, but it's not for all of the fish that we have. So we have the proportion of male and female in grills, but we wouldn't for these fish that were that biological data was collected and then the fish were released. Yeah, but with that said, it would be possible to use DNA technique in order to get at that question in future samples.
Yeah, you're probably aware that about 90% of the grills here are male, and probably 60 to 70% of the large fish are female. Just in closing, I'd just like to let you know that we'd be pleased if you want to do that type of research on the Marguerite. The Marguerite Salmon Association would be happy to provide you with some support here, some field workers, some technicians. We're always interested to have you down here, and we're always interested in science.
Thank you, Bill. Your collaboration has been really, really great over the years. Like, you're a great collaborator. And with that said, we have analyzed some of the scales for Marguerite. So we have scales from 91 to 96 that we have measured, and that's going to be the next step to actually look at potential population level difference. We want to compare Miramichi, Marguerite, and Two Rivers in Quebec to see if we're seeing the same pattern or if it's just specific to the Miramichi River.
Thank you.
Thank you.
Okay, we have Alex Belak is going to ask his question. And it doesn't seem to want to unmute. Oh, Alex, you're muted on your end. Would you please unmute? While he's doing that, I'll move on to Christiane Dufresne. Okay, Christiane, go ahead.
Yes, hi. Hi, Cindy. Excellent talk. Really, really interesting. Thank you very much. Just a question regarding the growth if I understood correctly, you said that growth decrease when they leave sea and initiate their return migration. Is that correct?
So on this scale, we can see for both age group that there's actually an increase in growth in the summer period and a decrease during the winter period. And then after winter, there's a pickup in growth that occurs for both age groups. But then at one point, there's a separation that occurs at about circa 30 to 40. And that's when most likely the sea winter fish are on their return migration.
Okay, so does that mean that when the fish migrate, they will not grow?
They will not grow. It depends. It could be that maybe they're not feeding as much and not growing as much, or that they're using a lot of that energy to actually migrate. So it could be one of the two.
Okay, great. Thanks a lot.
Thank you.
Okay, I'm going to try Alex here again. Alex, you were muted on your own computer. If you could unmute yourself. In the meantime, I'll move on to Craig Purchase.
Yeah.
Okay, Craig, go ahead.
Am I coming through there now?
Yes.
Okay. Hi, Cindy.
Hi.
More of a comment than a question, something to make you aware of that might be useful to you. 10 years ago, I had a PhD student who was doing some work in Ireland. This is not published, but it might be available and to be useful as part of the picture. There on the Berries Hole system in Mayo, it was a similar question about what causes a fish to turn around after 1 year to come back versus one to come back after 2 years. But the unique thing about that project that's a bit different than what you have here, of course, you have data on what the 2 sea winters and 1 sea winters did in terms of growth, but there might be other things that are different about them in their freshwater histories or whatnot. In this project in Ireland, all of the parents of the fish were known. They all had 2 sea winter parents. They were spawned in the hatchery system there in the Berries Hole. They were raised in captivity in their freshwater life cycle. So their heritage, they all came from 2 sea winter parents. They had identical freshwater experiences. They were coded wire tagged when they went out. And about 2/3 of them came back after 2 sea winters like their parents did, but about 1/3 of them came back after 1 winter as a grilse. So the project was related to, well, was there anything, was there a pattern? And, these were basically siblings. So with identical freshwater experiences to their other siblings. And so that's exactly what we were doing, was looking at from the time they entered the sea to the winter minimum and after. So it was a similar type of thing, but kind of controlling their freshwater experience type of thing. But it's never been published. So if that stuff is at all key, I could probably put you in contact with the holders of that information or whatever, and because it could be an interesting add-on to the story or something.
Oh, that's an interesting study for sure. Do you recall if they looked at sex of the fish in that study?
I'm sure we did, because they would have actually handled all the fish type of thing. And the student gave some presentations on that. It was never published, but I'm sure I have the PowerPoint slides or the poster or something like that. I'd say it was 10 years ago. What it was the exact nuances I'm a little bit rusty on. But anyway, that information does exist.
Mm-hmm. No, it's interesting to see that they were grown in a similar freshwater environment, and yet you see that that separation that occurs, although they are from the same parents.
Yeah, so related to, I think, exactly what you had, but with, I guess, for lack of a better word, a bit messier data in terms of what they did beforehand. And that the idea is that the ones that enter the sea and grow the fastest are more likely to turn around and come back as a grilse than, I mean, that exactly fits with life history theory, and faster growth rate should be earlier age at maturity. So the idea is when they enter the sea, from the same genetic background, the fastest ones should mature quicker. In other words, turn into a grilse and come back, and the slowest growing ones would delay maturity for a year.
That's correct. Yeah. Oh, that's great. Thanks. I might reach out.
Yeah, just reach out to me and I can follow up with you.
Okay. Alex has switched over to a written question because he's having some difficulties unmuting on his end. His question is, he says, it's an interesting presentation. Can you please tell us what practical application this study might have with respect to managing salmon stocks?
Yeah, it's an interesting question. So I guess this study is trying to contribute part of the puzzle of why we are observing low returns to rivers. So we're trying internationally, there's people trying to understand what are the drivers of low returns. It's likely multi-factor. However, one of the hypotheses is to try to understand if there's any changes in growth that occurs. So it doesn't, although it doesn't have like a direct fisheries management application, it's not gonna change any of the management actions. It's if we know what could be at play in terms of drivers, while there may be some opportunities to further that understanding and then perhaps act on it. Now, I do realize that if it's related to growth in the ocean, it's very difficult to do any action in the ocean. However, if, for example, if we would have seen any changes in growth occurring in the southern Gulf of St. Lawrence, perhaps there could have been something closer to the river and actions that could have been taken. So really that's what it is, trying to get a handle on whether growth has changed and if it's more of a ocean change or whether it might be occurring closer to home. We're also looking at freshwater growth as well, so it could have been something in freshwater that we would have detected a change. So yeah, hope it answers your question.
Okay. Thank you. The next question is also a written question. It's from Pedro Nilo. Thanks for the excellent presentation. What is the biological interpretation, if any, of a circuli? Is it time related or energy related? And is there any lag observed between the circuli itself and the conditions of growth?
Yeah, so that's a good question. So there has been some control experiments that have looked at when circuli are deposited, and it's and the estimation is something like every 7, 8, 9 days, there's a deposition of a circuli that occurs. So it's a relation in time. That's why when I had this slide about the winter minimum, so that study by Carlson where they showed that the winter annulus occurs somewhere between mid-February to mid-March, that means that when the growth picked up again and separation, divergence of the two groups occurred, maybe sometime like in April or May. So it's just putting that back in time, and it's really consistent through the time series. So it seems to indicate more of a time aspect. Now, in terms of scale, growth in relation to body condition, although it's a good predictor with a lot of noise around the data, it's a good predictor of fork length sometimes. However, I think body condition is a totally different question because you could have two fish of the same length. However, one could be very skinny and the other one could have a higher body condition, so it could be chubby, for example. So I'm not too sure what the link is between growth and body condition per se. It might be a harder link to make. But in terms of linking it to length of the fish, it seems to be reasonable to infer growth from scales to look at the link to length. Hope this answers your question.
Okay. So the next question is from Gerald Chaput. Thank you, Cindy. Can you comment or speculate on where in the North Atlantic the two age groups would be at the point where they diverge? And how far can they be for them to return to Miramichi by the time they were sold?
Yeah, it's good that you mentioned speculation, because maybe others have a better idea about this, but I would think it would be somewhere in the Labrador Sea to allow them to actually migrate back. If we assume that the divergence occurs like in April, May, for them to come back, we see them in river as early as June, July. So for them to come back at that time period, then it would have to be somewhere in the Labrador Sea, maybe towards the southern end of it, I would presume. But yeah, I'd be interested to see what you think about that.
Okay, so I'll just give it a couple more seconds. We don't have any other questions on the board right now. Okay, so that seems to be the questions so far. If folks do or have more questions later, please feel free to send one of us an email at FCAS, and I'll pass it on to Cindy, or you can email her directly. She's provided her email address there on the screen. Thank you very much, Cindy. That was a great presentation. We've recorded the session, and it will be available on our YouTube channel. Plus, I do want to remind everybody that our next webinar will be next week on the 11th of December, Stable Isotopes and You, a Primer for Environmental and Ecological Research by Dr. Bobby Nakamoto, Science Manager at SynLab. So a big thank you again to Cindy, and also to everyone who participated. We hope to see you soon. Thank you.
Have a great day.